Hi, I'm Heather James, conference producer at Technology Networks, and I'm here to introduce our next talk, the Leukemia and Lymphoma Society, Impacting Cell Therapies for Blood Cancer, from Basic Research to Clinical Practice and Patient Support. I'm pleased to welcome Dr. Laura Gruenbaum. Laura is the Vice President of the Therapy Acceleration Program at the Leukemia and Lymphoma Society, a philanthropy program aiming to accelerate high-risk, innovative blood cancer therapeutics. Law joined LLS with more than 20 years' experience in drug discovery and clinical development, including roles as Vice President of Biology at Gotham Therapeutics, Executive Leadership at Applied Biomath, and roles of increasing responsibility at Boehringer Ingelheim and Roche Pharmaceuticals. A warm welcome to you, Law. Following the presentation, we will also have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. Without further ado, I will now hand over to Laura to begin her presentation.
Thank you, Heather. I appreciate this great introduction, and good morning, good afternoon to all of you joining us online. I am really excited to be able to participate in this event and to share with you really a larger story, how a nonprofit organization such as the Leukemia and Lymphoma Society has been able to shape not just basic research and clinical development for novel cell therapies, with the example being CAR-T, but really also been critical for making sure that these novel therapies ultimately reach patients. So it'll be a little bit of a bigger picture story. And I want to start out by just introducing you to our organization a little bit. We are the largest nonprofit organization dedicated to creating a world without blood cancers. Our mission is to achieve cures, and you see here we are very inclusive in terms of including leukemias, lymphomas, Hodgkin's disease, myeloma, as well as a range of very, very rare indications in the blood cancers. And we do this, as I said, not just through funding research, but also to provide through providing education and critical support to patients, to survivors, to caregivers and families and healthcare professionals. And here in the US, we also have a very strong effort to drive advocacy for policies that can protect patients so that they gain access to life-saving treatments and they gain them at an affordable price. So not all of you, of course, are familiar with blood cancer, so I just want to give you a few numbers to put this into perspective. In the US alone, where I'm based, we have over 1.6 million people living with blood cancer or being in remission from one of the major blood cancers. Approximately every 3 minutes, someone here is diagnosed with blood cancer. And sadly, to the present, about 30% of blood cancer patients still do not survive 5 years after the diagnosis. And the last tidbit here, that is also something we're very concerned about at LLS, about 40% of all pediatric cancers are actually blood cancers. And some of you may already know that, yes, CAR-T therapy has helped, and many kids have a very good prognosis, but there's still a lot more to do. And we have a special initiative, the Dare to Dream initiative, that addresses that. So let me give you a little bit of the historical context here, how LLS has enabled and accelerated CAR-T. And what I'm showing you here is a very rough timeline. It's by no means complete. To just give you an idea how cell therapy, how immunotherapy and cell therapy generally falls under immunotherapy has developed. And this really goes back all the way, believe it or not, to 1891, when William Cooley was initially experimenting with bacterial toxins for the treatment of cancer. So he's really regarded as the father of immunotherapy. Obviously, those approaches are not what we're using today, but it really laid the groundwork for the initial recognition that the immune system and the immune cells could be really harnessed to fight cancer. What you see in green then are some of the milestones in our organization, and our organization contributed to the development of novel therapies, first and foremost, by also really supporting critical basic research and then translational research. So key milestones, of course, were the introduction of bone marrow transplants, first in the autologous setting, and then also in the allogeneic setting from a donor to a patient based on match. This was where our funding became critical already in the 1970s. And then, of course, after the cloning of the T-cell receptor, focus on really leveraging T-cell properties and the T-cell receptor for therapies really began. You can see, again, we were quite active already with early grants. And one point here I want to highlight, and I'll come back to it, in 1998, we actually provided grant funding to Dr. Carl June, who many of you may be familiar with, who was really critical for really advancing towards the first clinical trial and the first successful treatment with CAR-T therapy. And this was at a time when government agencies here in the US were actually not quite ready to support this work, and we were. And the success story, of course, has continued. These therapies have been improved significantly. And then ultimately, in 2017, we had the first registrational trials and the first FDA approvals. And we've seen many, many more, as I'll show you, following on this. And I'm highlighting here a key milestone. We've actually contributed directly to a partnership with a company called Kite Pharma to one of those first two. So all in all, since 1998, we have invested over 50 million in CAR-T for leukemia, lymphoma, as well as multiple myeloma. Now, I assume this audience is already quite familiar with the concept of chimeric antigen receptor T-cell therapy. So I'll just give you a very brief summary here. Of course, the basis of this concept is that we are going to engineer a T-cell, a tumor-killing T-cell, to better enable it to really effectively kill a tumor cell. And what we're leveraging in this case is actually a really interesting construct that differs from what you see in the middle here, which is, of course, a native T-cell receptor as it's expressed on T-cells, which is a very complicated structure. Let me just make sure this. There we go, which is a very complicated structure. And with a key feature really being that antigen recognition via the T-cell receptors really depends on something we call HLA, antigen presentation in the context of HLA. And T-cell receptors, of course, recognize both intra as well as extracellular antigens. Now, on the right-hand side, under B and C, you see two examples. I'm using here schemas that represent the two first approved agents, Yescarta and Kymriah. And what I want to highlight here just briefly, again, is that the CAR construct is really a composite combining the antigen recognition domain, we're seeing this here as SC3, from antibodies with transmembrane domains and co-stimulatory domains, such as CD28 and 4-1BB domains that are actually part of the co-stimulatory complex in the native T-cell receptor. And then with the intracellular signaling domain, the TCR zeta domain that again derives from the TCR. So it's really a composite that we're using here and that we're introducing in T cells. And the beauty is, of course, that by using the antigen recognition domain, we can specifically target antigens that are highly expressed, overexpressed on tumors, and thereby specifically direct these T cells to these tumors. So again, just a brief primer for those of you who are not yet as familiar with the concept. So the beauty of this approach is that at the present time, we have been able to achieve long-term disease control in a number of leukemias, multiple myeloma, and many lymphomas for patients who really have exhausted all the options. So one other thing I want to tell you about CAR-T, the introduction of this therapy has really significantly changed and also challenged physicians as well as patients, because CAR-T is not really unlike any therapy that physicians and patients have been familiar with before, mostly chemotherapy, then some targeted precision therapy with small molecules. CAR-T, and it's depicted here in the schema, involves a complex multi-step process that really starts with the collection of a patient's leukocytes by apheresis. And this then initiates actually sort of two parallel work streams that are happening. One is focused on the ex vivo processing of T cells, and this involves steps such as T cell activation, transduction with a CAR construct. There are a variety of approaches, both viral, mostly replication inactive retroviruses, lentivirus, for example, electroporation. We are now seeing lipid nanoparticles, etc. So a range of approaches to introduce the CAR. And then a third step is actually the expansion of the T cell population, because you do want to introduce a good number of T cells, of these modified CAR T cells, back to the patients to effectively fight the tumor. Now, at the same time, our patient, of course, is continuing to suffer from cancer, and sometimes rapidly advancing cancer. So a patient may require something we call bridging therapy to help manage cancer, and then ultimately needs to receive a lymphocyte depleting chemotherapy, because we've discovered that it is important to reduce the lymphocyte population, so that after reintroduction of the CAR T population via infusion, those CAR T cells now have somewhere to go. They can expand, they can proliferate, and they can stay. So a complex process that cannot just be done at any community hospital site, and I'll come to that in the second part of my talk. A lot of education required for patients, for physicians, a lot of learning how to manage these novel therapies and aspects of safety as well. So the very first CAR T target, I'm showing it to you here, it was called CD19. So this is a surface antigen highly expressed in lymphoblastic leukemia and many other B-cell malignancies. And it was selected because, and you can see it in the schema, it is expressed on B cells and many of their precursor cells. And that's where a lot of those malignancies of B-cell lymphomas derive from, but it's actually not expressed on bone marrow stem cells or adult tissue. Or many other adult tissues. So that gives it some selectivity, which is really essential for a therapy to be tolerated by a patient. So what I'm showing you here are some of the data that were actually critical supporting the approvals of the first two CAR T agents. And you see here examples again for Kymriah as well as Yescarta, those first two agents on the left-hand side, and relapsed refractory B-cell acute leukemia on the right-hand side, data in a range of different aggressive lymphomas. And you can see that what you actually achieve are remissions, and you can see that some of these remissions, we're estimating maybe around 40–50% are really long-term remissions, which really raises the hope that we may really consider some of these long-term remissions cures. Most of you are probably aware we're always very careful with the term cure because it has such an important and specific meaning. But we're coming to the point, and I'll show you a couple of vignettes where we think that these could really be potentially considered cures for some patients. So let me tell you a little bit more, picking one of these first two agents, Yescarta, you see it here on the right-hand side, developed originally by Kite Pharma, and tell you how we supported the development of this agent. You can see on the left-hand side, this was for very aggressive non-Hodgkin's lymphoma. So this is a range of B-cell lymphomas. And let me again remind you of what it actually takes to bring such a therapy all the way to an approval. Started out again 1998 with our initial funding for CAR T that supported actually both Kymriah as well as Yescarta, and ultimately many of the other therapies and indication and approvals that relied on these downstream. In 2015, we started supporting this therapy with our therapy acceleration program. This was a really novel approach in venture philanthropy to use partnerships and funding for pharma companies or small biotech companies to really help incentivize these companies to develop novel therapeutics, and to also in this partnership really offer all the expertise, the experience, the knowledge, the network that we at MLS have available to help them develop the therapy efficiently, quickly, and really for the patients who could benefit most. So this has really been the goal of the program. It's been a very successful program, has led to 3 FDA approvals, as well as inclusion in the NCCN guidelines for an additional drug, and is currently supporting, also has 2 assets that are in registration of the trial. So quite an interesting and successful program. So through this program, we've supported the clinical trials. Clinical results were presented in 2016. Very exciting clinical results that ultimately led to 2 events. Kite was acquired by Gilead, and in October 2017, FDA approval occurred. The first FDA approval for the therapy as well as roughly little less than 3 years later, a new formulation was approved for an additional indication. Another approval then in 2021, again for Yescarta, and we are currently seeing trials for a range of additional indications as well as earlier lines of therapy. So a drug that continues to be explored to further widen really its application in blood cancers. Now, I'm really happy to report that since 2017, 12 CAR T approvals have been achieved, and I'll not go through them in detail. But one of the things that I'll highlight here is that first in 2021, we started to see approvals not just for leukemia, acute leukemia and lymphoma, but also for multiple myeloma. There are 2 approved therapies out there. You can see them here. It's Abecma and Carvykti. And those have very significantly also impacted patients with multiple myeloma and really improved their therapeutic options. So at this point, maybe let me just stop for a second and just give you a vignette, 2 patient stories to highlight a little bit of where CAR T has gone and where it's going. So this is actually Emily Whitehead, the very first patient who was treated with CAR T therapy. This happened in early 2012, I believe. Emily was 5 years old, and very first treatment, much to learn, very challenging. But Emily has been cancer-free since 2012. She's doing well. She is a student at UPenn here in the US, and a wonderful advocate also for cancer patients and for the importance of novel therapies. And now a second example. This is Lori Adami, also very active patient advocate and CAR T patient and follicular lymphoma survivor. She's been cancer-free since 2020. And again, these are amazing stories that illustrate how this is affecting individual lives. And these are obviously people who also help us tremendously to raise funds and help us support more work, because we still need to do way more. As I showed you, we have maybe 40 to 50% of patients who are long-term cancer-free, but there's a lot more to do for all the other patients who do not achieve these types of things. Okay, so where have we gone from here? Because I would say we really have only explored the tip of the iceberg so far. There are next generation CAR T cells actively in development, and there are many things that, of course, we learned that could be improved over these first approved therapies, whether it's around selecting the most active, the most longest lasting T cells, the most robust T cell, optimizing and really accelerating the manufacturing process so that we can include more patients in trials, because sometimes patients with rapidly progressing disease were not able to wait for the production, for the manufacturing of their CAR T cells, or the quality of their own T cells might not have been good enough. And that latter issue could be addressed with off-the-shelf allogeneic T cells that are produced from donor populations, and that could basically be available immediately when a patient turns out to be a candidate for CAR T therapy. Reducing toxicity has been really important. I think the medical community has learned a lot how to manage toxicity. I'm just mentioning here two primary concerns: cytokine release, as well as neurotoxicity, that both in the worst case scenario can even lead to the death of the patient. So both in terms of mitigating this with appropriate pre-treatment, with immunosuppressive agents, with anti-IL-6 for cytokine release, for example, has made a big difference. And engineering can also, to some degree, address these toxicities. We've learned that already. And then there's, of course, much more engineering to armor CAR T cells to make them even more powerful, to help, increase, maybe allow them to also address multiple targets, not just one target on a cancer cell, many approaches finally. And then last but not least, I already showed you how CAR Ts moved from acute leukemia and lymphoma to multiple myeloma. Many more targets and blood cancer indications are being explored as we speak. So we hope to see more success stories there in the near future. So I'm a bit too fast. So what I want to show you here is, again, just to highlight what we've done in this space. And what you see here in red is all the funding that we have provided to academic institutions all over the world to develop CAR-T therapies. And then the other piece that I'm focusing much less on today, but I wanted to at least mention it, is that we're also funding other adoptive cell therapies. These could be NK cell therapies, they could be myeloid cells, other types of infused leukocytes. So you see, this is really an effort that's been ongoing since the early 2000s, and that we really continue to fund and support heavily, because we believe that there's much more potential for patients. So let me give you, again, a couple vignettes of what our venture philanthropy program, the Therapy Acceleration Program, is doing. And what I'm highlighting here is a company, Caribou Biosciences, actually co-founded by Jennifer Doudna, who was one of the co-recipients of the 2020 Nobel Prize in Chemistry. And this company is pursuing an allogeneic CAR-T and NK therapy approach. So I'm just giving you here an example of what they're doing. They're actually using precision genome editing with the next generation CRISPR technology that allows to armor CAR-T cells to basically make them more robust, more active, longer lasting, and also to engineer them to prevent and reduce immune rejection, which has been one of the big challenges in the allogeneic space. They actually at the current time have 3 active clinical trials for 3 different agents, and I'm listing for you here. So again, we're looking still at improvement for patients with non-Hodgkin's lymphoma, multiple myeloma, and we're really excited about the 3rd trial here, also for acute myeloid leukemia, where survival is, prognosis is really quite bad for patients, and we have not yet seen any success for CAR-T or any cell therapy. And the second vignette, this is a company earlier stage, but also something we're really excited about, a company that was actually co-founded by Marco Ruella at the University of Pennsylvania, a recipient of Alalas grant funding, and grant funding has definitely contributed to Marco's work and ultimately also impacted indirectly what Victoria is doing now. So they also have a novel CAR-T platform, and it's a very unique approach where they're knocking out a surface molecule in T cells called CD5. And what this knockout does is actually does 2 things. It increases the anti-tumor efficacy of T cells, and they've already shown this in many liquid and solid tumor models. And it also prevents something that we call fratricide, which is a CAR T cell based on CD5 being expressed on the tumor cell and the CAR T cell, also other CAR T cells. So that was something that had really limited the application for T cell lymphoma. So the company is developing their current program to address unmet medical need in T cell lymphoma, another indication that has very, very poor outcomes and really insufficient treatment options. So I'll open up the world even more, and just a snapshot, what's next? So we're of course seeing CAR-Ts being explored in solid tumors, based on the initial approvals they're learning in blood cancers, and I won't go into detail. I think you can see it clearly on the slide. Many solid tumors that also still lack good treatment options, last line therapy for patients. There are over 1,000 patients that have been in trials and over 500 trials. And then there's work ongoing to really explore what this approach can do beyond oncology, be it in the infectious disease space for HIV/AIDS. A lot of excitement and many interesting publications coming out for applications for autoimmunity, organ transplant. You can see the lithography on the right-hand side recently published in Nature that shows how the number of trials has been increasing, in particular for lupus, a severe systemic autoimmune disease. That significantly reduces life expectancy and quality of life, as just a very prominent examples. And then, of course, going even beyond that, looking at heart failure, looking at fibrosis, and also exploring in vivo approaches to basically generate CAR T cells in vivo in the patient. And that is really probably the ultimate frontier for these types of therapies. We're not there yet, but we're hoping to. And the trials are expanding worldwide, which is really important as well. You can see it here. Started in the US in 2006, more than 800 trials. Numbers probably changing already today as I'm speaking. And we see a lot of work happening in China, many companies, many trials as well. So definitely something to watch for us, and looking forward to hopefully more advances in this. Okay, so for the last part of my talk, let me just switch gears a little bit. And what I'm showing you here is what we at LLS call the circle of care. And the circle of care to us really means that our work cannot be impactful unless we address every single aspect, every single piece in the circle that ultimately is needed to bring novel, better therapies all the way to the patients who need them. So I've talked to you about research funding. I've not talked to you about career development, but that's another important piece. Basic and translational research, clinical trials. And now I want to talk a little bit more about the other parts of the circle, which are really focusing on what happens after a drug gets approved. So very challenging to bring this therapy to the real world. We need to educate patients and physicians. We have to improve access. I already told you that these therapies are complex, and they generally are administered and offered in large medical centers, and not every patient is close to the large medical center. We need to match patients with appropriate clinical trials. We need financial support to make these therapies affordable. And here in the US in particular, we need to improve insurance coverage, reimbursement, so that patients don't go bankrupt over treatment such as that. So I'll just walk you through a few of our resources, and I really encourage you to take a look at them and to connect with us if you're interested in learning more. A wide range of specific educational and support resources directed at patients, at caregivers, at healthcare professionals. And in particular, we have the Patty Robinson Kaufman First Connection program, which actually connects a patient who is considering CAR-T therapy with another patient who has undergone therapy and really can share their experience. Something our patients have told us is extremely valuable. We have an information resource center that is basically available 24/7 for virtual support or via the phone as well. And that can provide one-on-one personalized information and advice. It's staffed by highly trained oncology social workers and nurses. And I'll tell you that these colleagues get in touch with us as a research scientist all the time to make sure we really bring the latest information to our patients. Again, check them out. You see the link is right there. And then one thing I'm really proud of is our clinical trial support center. These are nurses who, again, work one-on-one with patients, sometimes over years, to help them find the appropriate clinical trial for them. They will work with whoever the patient feels comfortable with to work with the patient's entire care team, not just their oncologist. They will call up the trial sites, they'll talk with the investigators, and also highlight to the patient any and all resources that can help the patient manage all the challenges with attending, participating in the clinical. And then we have financial assistance, and I'm just highlighting one of the many financial assistance programs we have, recognizing the challenge of helping patients actually get to academic centers where CAR-T therapy was offered. We instituted the Susan Lang pre-CAR-T cell therapy travel assistance programs. And this program helps with transportation and lodging expenses for patients who will be evaluated to receive CAR-T. And I can tell you that this program has been incredibly effective. We did a survey in the spring of 2023, last year. 93% of patients who were awarded one of these travel assistance awards reported back that they were able to complete the pre-CAR-T evaluation process. And out of those patients, 95% successfully enrolled in CAR-T therapy, either with an FDA-approved agent or on a trial. So this is amazing. This is an amazing result. We also heard back that 12% of patients had to travel more than 8 hours to get back to the academic centers. 10% had to travel 5 to 6 hours, 17% 3 to 4 hours. So that just gives you a picture of how challenging it can be and how critical this type of support is to really help patients. And last but not least, again, here in the US, improving access via policy change. is really important, both at the federal level as well as at the state level. I'm highlighting here two success stories that LLS advocacy and the Office of Public Policy really contributed to: the Clinical Treatment Act, that really led to mandated Medicaid coverage for routine care that a patient receives while enrolled in the trial, and the Medicare National Coverage Determination that established CAR-T as a covered benefit for Medicare beneficiaries nationwide. So this actually captures a lot of the retired elderly patients that really are impacted by blood cancer. But of course, there's a lot more to do. We need to improve commercial coverage to ensure out-of-network access. Again, one of the intricacies of our US healthcare system, provide better access out of state to providers and facilities, especially for children who rely on Medicaid for coverage. And a very large percentage of children do rely on Medicaid, unfortunately. So we're currently advocating for the Accelerating Kids Access to Care. And we'll continue to do this because, again, there is much more to do to really ensure that the latest therapies reach patients who need them. So at this point, I want to close, and I want to thank you for your attention. Putting here just a few numbers so you can appreciate hopefully the impact that we've had. We'd love to be in touch with you. Please check us out on our website and learn more about what we do. And I'm thanking you. I'm ready to take any questions.
Lovely. Thank you so much, Laura, for a fascinating talk. I'm sure that all our viewers will agree that was a really clear story of how LLS has supported the CAR-T therapies and hopefully can do so for future therapies as well. So as with all the talks, we're going to have lots of questions. I can see that we've got a few, but just a reminder that do keep putting those in the question box to the right-hand side of your player, and we'll get to as many as we can. So I'm going to kick off, Laura, with one of the questions, which is, what were the key challenges in the initial development of CAR-T therapies?
Yeah, that's a great question. And there were really a number of challenges, starting with the fact that oncology had, and blood cancer, of course, when oncology had really been shaped by chemotherapy. And learning about the immune system and understanding the contribution of the immune system in the context of blood cancer was a real challenge. And I think initially also for physicians to understand what happens when you unleash that power that lies in our own immune system, in our T cells, when we now equip them via CAR-T therapy to really be aggressively fighting cancer, understanding what that meant also in terms of the side effects, the tolerability. That was, I think, a very novel and tough and challenging learning experience for clinicians and for clinician researchers, and really identifying the best pre-medications and also particular risk factors that would put a patient at high risk to experience some very significant side effects, which can send someone definitely into the ICU. That was one of the big challenges. And then really helping the entire community of physicians as well as patients understand. I showed the slide earlier in the presentation, what that process looks like for CAR-T therapy, to really understand the process, to really understand what is happening, because this was really so different from any type of therapy that patients and physicians had seen before.
Lovely. Thank you so much for that answer. I know that with any sort of therapy for any area of patient treatment, it is always a learning curve. So it's really interesting to hear your take on
Absolutely.
that. I will pop to another CAR-T one. I can see lots of questions coming in, but just to follow up from that, how broadly are those CAR-T therapies being used now, and are they accessible to patients in the US and in other countries?
Yeah, so this is one of the things that we're very actively working on. You can actually look at a map that shows where in the entire world CAR-T is being used, not just in clinical trials, but is really an approved therapy that's being deployed in hospitals. And obviously, this is quite uneven. You can certainly see that in North America, European countries, it is much more accessible generally. But I can also give you an example just within the US. And that's really based on the fact that CAR T therapy is complex, as we just discussed, and it is generally being administered in large hospitals, academic hospitals, where you have the range of experts available, the capabilities in intensive care units to deal with situations that can arise potentially with the CAR T therapy. The ability to participate in the process, the production, the manufacturing, etc. So the bottom line is that many of the patients who need these therapies, of course, don't necessarily live very close to these academic centers, and as the basis of that, we're seeing that access is quite uneven. And I think this is where all the other activities within LLS come into play that are outside of funding research and funding clinical development and partnering with companies to really ensure that we find ways to bring patients to these therapies and to also make these therapies, really transform these therapies so that they can be deployed in community hospital settings, in rural settings, and that they are more affordable also from a cost perspective. This is something we're clearly struggling with in the context of many complex cell and gene therapies. The expense, the access for these therapies is something that I think is going to require decades of investment to come.
Absolutely. Thank you so much for that insight. Now, I can see that we've had a question come in, and this might not be one that's directly within your field of expertise, but I know that you have a good overview of sort of therapies on the horizon. So Amani has asked, what about using enzymes as anti-cancer? Is that something that you've heard about? Is that in the pipeline or not really in the scope at the moment?
Yeah, so that's an interesting question, because certainly leveraging enzymatic activity is something that is really being considered. And one example would be in the context of actually turning an active drug either on or off, because one of the general challenges that we have in blood cancer and in cancers overall is that some of the most active medicines that we have, unfortunately, will also target a lot of healthy cells and organs. So there's quite a bit of work ongoing to develop therapies that can be locally turned on and turned off. And some of these approaches focus on really leveraging enzymatic activities such as extracellular local proteases or activities in the lysosomes of cells to selectively activate or inactivate powerful therapies, and thereby, make sure that they get activated in the context of cancer and not in the context of a healthy cell environment.
Excellent. Thank you so much. That's so interesting. I wasn't aware of those ones. So hopefully that answered your question, Amani. Another question that we've got is, can we expect to see additional approvals in this space in the coming years, do you think?
I am optimistic. I think we can. And in my talk, I highlighted one of the areas where I think we might see an approval in the coming years, and that is the allogeneic CAR T therapy. And again, the big benefit here is that the source of the T cells is not the patient, him or herself, it's a donor. So these are healthy T cells from a donor that, again, haven't undergone what a donor has potentially, what a patient potentially has experienced, chemotherapy, many treatments, and the effect of cancer itself on T cell, the quality of T cells. So it's actually leveraging T cells from a donor that hasn't undergone all these treatments, and then really armoring them so that they can fight cancer and introducing them into the patient. Now, as you can imagine, any kind of allogeneic therapy has particular challenges associated with it, because there's going to be an issue of immune rejection. But we have very, very promising approaches in the clinic from a range of companies, including the company I mentioned earlier, that seem to be quite effective at reducing these types of liabilities. And these allogeneic CAR T therapies, I think they have the potential to help many patients that could benefit, but that are not able to access these therapies today, either because their disease is progressing so rapidly that they don't have the time to wait for CAR T production from their own cells, or because their own T cells are just not healthy enough to really be able to form very effective CAR T therapy for themselves. So I think we're going to see approvals in that space. And we're going to see other therapies building on that, hopefully also in additional indications.
That's definitely good to hear. And we have had a question that, funnily enough, seems to be related to that come through whilst you were speaking as well. So, Francisco, and forgive me, this isn't my area of expertise, so I might not be able to pronounce some of these things, but I will try my best. So, Francisco has said in autologous CAR T cell therapy, failure of long-term response might be related to immunosenescence or cellular aging of the CAR T cells coming from patients that have undergone several treatments. In the case of allogenic CAR T therapy, do those cells undergo quality control regarding the senescence?
Yes, that's a great question. And the first answer is yes, of course there is quality control, but, and that's something that we are all eagerly waiting to hear, things still happen when you introduce a cell into a patient and into the tumor environment. So the jury is still out on the long-term persistence of the positive CAR T effect, of the efficacy, long-term remission. Hopefully at some point we'll be speaking more about, again, patients who are cancer-free. And that only time will tell in the clinical trials. I think we're seeing very promising signs, and we understand a little bit more how we can modify CAR Ts to make these cells more robust, to enhance the chances that they will be persistent. But at the end of the day, only time will tell how good these therapies are, and that's what we're waiting for.
Perfect. Thank you. I hope that answered that for you, Francisco. And we are swiftly running out of time. So I think a very brief one to end on for you today, Laure, is what activities are the LLS focusing on at the moment for the next generation of cell therapies?
Yes, and there are a range of activities because, again, our universe within our circle of care, within our mission, is really touching on everything, not just on research and development and clinical development. So we are looking at other cell approaches. We're looking at novel approaches that are leveraging RNA-based approaches, lipid nanoparticle-based approaches, and really opportunities to bring cell therapies to a wider range of blood cancers. We've seen impact, as I mentioned earlier, in multiple myeloma, in addition to acute leukemia, some types of acute leukemia, and of course lymphomas. But there's much more to do, for example, in acute myeloid leukemia, where we haven't seen success yet. And then we will continue to work on really helping with access for patients, with education for providers, so that we can really maximize the value of what we have approved today.
Excellent. Thank you so much, Laure, for that talk and for answering all of those questions. I know that we got through quite a few there. Unfortunately, that's all the ones that we have time for today, but please do continue to send in your questions after the event. This event and this talk will be available on demand for the next 30 days to our viewers. So we will send any further questions through to Laure, and she'll be able to get back to you on those. So that just leaves me to say thank you again to Laure and to our audience for joining us, and we'll be back very soon with the next talk.
Thank you.